So I'll take a short moment to do an introduction, and then we can all listen to your talk. So today we have the pleasure of having Adam Kohn, who is definitely an authority when it comes to visual neuroscience. Adam received his BA in physics in UC Berkeley, and he has a PhD in neurobiology in year 2000. He did the postdoctoral training with Tony Mopshin in NYU. And from 2006 till now, which is maybe more than 15 years, he is in Albert Einstein College of Medicine, and currently a professor in the Department of Neuroscience, Ophthalmology, Visual Sciences, and Computational Systems Biology. Dr. Kohn's research focuses on issues of population coding, plasticity, and cortical communication in the visual cortex. I think his title today is about this cortical communications, which we're excited to listen to. And he uses a variety of techniques, including neurophysiology, computational neuroscience, and perceptual methods, careful psychophysics to address these issues. And with that, I basically give the floor to Adam, so we can listen to his talk. Thank you.
Thank you. Thank you very much. You can hear me okay and see my screen?
Yes, I can. Okay. We can hear you. We can see your screen.
Okay. So thanks very much for the introduction, and thank you for the invitation. I wish, of course, I could come and give this talk in person and attend the meeting fully, but I'm glad anyway to be able to tell you a little bit about some of the work that's going on in my lab. So because of the format, it's a little more difficult to interact, but if there are questions during the talk, please feel free to ask them or post them in the chat box. I'll try and keep an eye on that. So what I'd like to tell you about today is our work on cortical communication. And this is a joint project among 3 labs. So specifically my lab, Byron Yu's lab at Carnegie Mellon, and Christian Macac's at Champalimaud. And the work that I'll be presenting today was carried out primarily by João Semedo, who is jointly advised by the 3 of us, based on some recordings that Amin Sanfakali had performed in my lab. Amin came from Tehran and came to my lab as a postdoc, and then afterwards as a student, and Anna Jasper, who's currently a postdoc in the lab. So to motivate our interest in cortical communication, I like to start by pointing out something that I think is common to most of us interested in neuroscience, which is understanding how organisms gather information from the environment and use those inputs to generate appropriate behavioral output. And one really simple circuit for doing that is illustrated here. So here's a really simple input-output relationship, which is the knee reflex arc. So in this circuit, what happens when you go to the doctor is if you get hit on the kneecap with a rubber hammer, it sends afferent information to the spinal cord through a synapse or 2, generates motor output, which causes your foot to jerk forward. This is a really simple circuit and a really boring input-output relationship because the output in healthy individuals is exactly the same each time the input is delivered. So you provide an input to the nervous system, you get exactly the same output each time. Fortunately, our brains are more flexible and interesting than this, and we are able to generate very different outputs given even the same sensory information. I think this is intuitive and obvious, but one really nice demonstration to make this concrete is recent work from the Newsom lab, which I'll present to you briefly here. So in this study by Valerio Monte, animals were trained to perform a discrimination task. So they looked at a computer screen. They were presented with a field of dots of different colors. And then after a delay, they had to indicate something about their judgment, their percept of this moving field of dots. The stimuli that were used are shown here. They actually varied on 2 stimulus dimensions. So the dots could vary in the color content, consisting mostly of green dots or of red dots, or different values in between. And the dots moved, so they could move to the left or to the right, and the percentage of dots moving in one of those two directions could also be varied. So this was a fixed stimulus ensemble, and the animals were trained to report either the color content or the motion content of the stimulus. So here's some behavioral data from one of the animals in this study. What's plotted here is how often the monkey indicated that the motion was moving to the right as a function of the motion content of the stimulus. So if there was a lot of rightward motion, the monkey always said stimulus is moving to the right. If there's a lot of leftward motion, the animal very rarely said motion to the right. And importantly, the animal's judgment in this task were not influenced at all by the color content of the stimulus. Now, the easy thing for us to do as humans, and the difficult thing that Valerio managed to train animals to do, is if the context of the task changed by the cue presented on the computer screen at the bottom of the computer screen, using exactly the same stimuli, the animal could generate a very different behavioral output. So specifically now, when the animal was in the color context, the animal's judgments were determined by the color content of the stimulus and hardly influenced by the motion content. So exactly the same pattern of stimuli, but generating very different patterns of responses depending on the task in which the animal was embedded. So I think a central question in cognition and perception is, how is it that using a brain that's more or less fixed anatomically and in terms of its synaptic weights, are we able from moment to moment to generate very different outputs using the same sensory information? And the dominant hypothesis in the field, which is also the motivation for our work, is that this type of flexible behavior requires flexible and selective signaling between different parts of the brain. So virtually any task involves activity distributed across a whole bunch of different brain areas. And the thought is, if we perform different tasks with the same sensory input, that's because we're allowing different parts of the brain to communicate with each other depending on the goals, our goals at a particular moment. So this is what we'd like to understand. We'd like to understand what are the mechanisms in the brain that allow for flexible and selective communication between different parts of the brain on a moment-to-moment basis. So, and that is going to be the focus of the talk today. And specifically, what I'd like to try and convince you of is a new way of thinking about how this problem might be solved in the brain, this problem of flexible and selective signaling. And what I'm going to try to show you are some new principles by which we think signals are routed between different brain areas. So for the majority of the talk, I'm not actually going to be focusing on behavior, but taking a step back and trying to understand what determines at any particular moment in time whether two brain areas are effectively communicating or not, and how might that be flexibly changed depending on the goals of the organism. So the outline of the talk, what I'd like to tell you is the following. I'll tell you, of course, a little bit about how we do the experiments. I'm going to introduce this concept of the communication subspace, which we reported on a few years ago. I'll tell you a little bit about how that's defined, the mapping dimensionality between areas, and the role of the target population and how it relates to dominant dimensions in the source population. In this part of the talk, I will get a little bit technical. For those of you perhaps less familiar with these concepts, I think this is generally a fairly quantitative, computational, and systems neuroscience audience. But for those of you less familiar, I'll take a step back and explain to you why we think this idea of communication subspace is really interesting and how it compares to alternative schemes in the field for regulating communication between brain areas. At the end, I'll spend a few minutes telling you about some work that just appeared a few days ago, looking at the dynamics of communication between areas, specifically feed-forward and feedback signaling, and how the communication subspaces between areas may relate to behavioral or decision variables. All right. So the experiments in my lab, and for the purpose of today's talk, are performed in the model system illustrated here. So in my lab, we rely almost entirely on electrophysiological recordings from macaque monkeys. And what's shown for you here is the visual system of the macaque monkey. So this is a side view of the macaque brain. Each area, each labeling here corresponds to a distinct part of the cerebral cortex. you can see that the visual system occupies a large part of the cortex, the occipital, temporal, and parietal lobe. On the right-hand side, you see the same thing, the macaque visual system, but now laid out for you as sort of an engineer's wiring diagram. So each box here is, again, a distinct cortical area. The black lines indicate an estimate of the number of axons that go between each of these different areas. And what you can see is that visual processing, involves a large number of different areas that are very richly interconnected. So if we think back to the Monte task that I mentioned a few slides ago, involving color and motion processing, we might expect that the animal's decision might rely on signals in one part of this system being relayed to decision areas in one context. And then a few moments later, we would rewire things so that other parts of the visual system would be communicating their sensory signals to the decision areas. Okay, so for the majority of the talk, I'm going to be focusing on a specific pairing of areas, namely V1 and V2. So these are the first two areas, the first two stages of the cortical processing stream. They're both very large parts of the cortex, so both V1 and V2 are about 10% of the cerebral cortex. And they're also very, very richly interconnected. So these are among the most dominant feed-forward and feedback pathways in the visual system, connect these two visual areas. How we do the experiments is illustrated for you here. So for the first part of the talk, I'll be focusing on recordings in anesthetized animals. We place the animal in a stereotax and we implant two recording devices. So one recording device is the Utah array. So this is a grid of roughly 100 electrodes. We implant this in the superficial layers of primary visual cortex, or V1. And then simultaneously, we lower a set of tetrodes, movable electrodes, through V1, through the white matter into V2. And we place these tetrodes specifically in the middle layers of V2. So what we have here is the simultaneous monitoring of an output population, that's the neurons that live in the superficial layers of V1, project out of V1 to V2. And at the same time, we're monitoring the neurons receiving those signals, which live in the middle layers of V2. And the neurons that we can record in each of these two areas are indicated here. In V1, we record typically 100 units, and in V2, roughly 30 units, where units refers both to single neurons and small multi-unit clusters. And we're going to be using both of those types of signals for the analysis today. All right, so of course you can perform recordings like this from many pairings of areas. There's a couple of advantages of doing these types of experiments specifically between V1 and V2. The first is these are very well-studied areas of the brain, so we know a lot about the connectivity between them and about the functional organization, both in V1 and V2. Second, we know that V2 is very dependent on V1, so if you were to lesion V1, you would eliminate most visually driven activity in V2. So that means that the activity we're monitoring in V1 is functionally very important for the cells we're recording from in V2. And the third advantage of this model system is that both V1 and V2, because they're areas in the early visual cortex, have really nice retinotopic maps. And that lets us place our electrodes so we can record from groups of neurons that represent similar parts of the visual field and therefore are likely to be communicating with each other. So that's shown for you here. This is actually data from one of our recording sessions showing you the receptive field alignment that we achieve. So in this plot on the right-hand side, each of the little blue dots indicates the center of the spatial receptive field of one of the V1 electrodes. And you can see that there's sort of a loose grid-like arrangement consistent with the grid of Utah array electrodes. The red dots correspond to the recordings with the tetrodes in V2, which are spaced linearly. And then the shading here, the light pink shading and the light blue shading, give you an estimate of the size of the spatial receptive field at the eccentricity we record. So Amin Zanfakli, who did these recordings, became very good at placing the electrodes such that we achieved not just overlap of the receptive fields, but also very precise alignment of the center-to-center spacing of the neurons in the two areas. And for much of what I will be telling you, that type of alignment is important for seeing the sorts of functional interactions that I'll be telling you about. Okay, So we have this arrangement where we're recording an output population in V1, an input population or a receiving population in V2. And now the question is, how are we going to study or try to understand the communication between these two parts of the brain? Now, of course, we're not the first people to ask that sort of question. And the way that people previously had tried to understand interactions between two brain areas is by recording from one neuron in one brain area and another neuron in another brain area and looking at the moment-to-moment correlations or coherence of the activity of those two cells. That was informative. We learned something from that, but of course, recording just one neuron from this very large group of neurons involved in both V1 and V2 in processing any visual stimulus gives you a fairly weak view of the potentially rich interactions at the population level. About 10, 15 years ago, there was a body of work that realized this and tried to get power in asking this sort of larger scale question by replacing activity of one neuron in one of the areas with recordings of the local field potential. So the local field potential is a low frequency extracellular signal that reflects in a complicated and not well understood way the activity of a larger group of neurons. And so you can study interactions between brain areas by relating the activity of a neuron in one area to the local field potential recorded in another area. And at the bottom of the screen are a number of high-profile studies that did precisely this type of recording. Again, these were very influential and helpful studies. They showed us that communication or the interactions between brain areas can change a great deal from moment to moment. But again, relying on the local field potential gives us sort of a poor summary of what the population activity is in the targeted area. And again, we're relying on just one neuron rather than understanding the complex interactions among neurons that may be important for determining how areas interact with each other. So what we wanted to do is really understand and tackle this question of between area communication using large groups of neurons recorded in the two areas and understand interactions at the population level. So then we face the question, which is, what is the right analytic framework or the right way of thinking about how to study population interactions? And this is where our collaboration with Byron Yu and Christian Macaque began. And what we decided is to approach this question of interactions through a lens of dimensionality reduction. So I think most of you know dimensionality reduction has gained a lot of traction in the field as a way to understand population responses within a single area. And we wanted to take that type of approach and use it to understand interactions between areas. So in this approach, we make use of the fact we record from many neurons in the two areas. We extract from those high-dimensional population recordings a smaller number of latent variables that summarize this activity. And then we understand the interactions between the areas as interactions between these latent variables. So there is a simplification here. We're making use of complex, high-dimensional signals and distilling them. But rather than relying on a signal like the local field potential that we don't understand well, here we understand precisely the mathematical relationship between the full signal and the reduced representation that we're going to create. Okay. So this is sort of the spirit and the philosophy of the approach that I'm going to show you. And to make it a little bit more concrete, let's consider, before we jump into the data, a little bit of synthetic or toy data. So here's some synthetic data. This is recordings from a hypothetical population of 3 V1 neurons. And each circle here, the activity of each neuron represents an axis in this population activity space. Each circle represents the response of that population at a particular period in time. And what you can see is that the distribution of activity is not random, but has a certain structure to it. And dimensionality reduction approaches applied to responses like this can extract the dominant statistical structure. So for example, if we apply principal component analysis or factor analysis, we can define a dimension that captures the dominant fluctuations within this population. We refer to this as a dominant dimension. So if we were to project each of these measured responses onto this line identified by PCA, we would have a summary of the activity of these 3 neurons with 1 number, which is the value projected onto this line, as opposed to the 3 numbers, which is the 3 neurons we started with. So this is the dimensionality reduction step. So this is great, and people like Krishna Shenoy and Mark Churchland and many others have used this type of approach to further our understanding of functions of activity within a single area. We would like, though, to understand, not to summarize activity within one area, but to understand the relationship between this activity and the activity of a downstream circuit. So in this hypothetical example, we've taken this population response and we've now shaded each trial by the firing rate of a hypothetical V2 neuron. And you can see that the activity again has some structure with respect to V2. And specifically, we can capture the relationship between activity within V1 and in V2 by performing, for example, regression and defining a vector which would allow us to predict the V2 firing rate. So now by projecting these responses onto this regression line, we would have a good prediction of how V2 will respond at any instant in time. And you can see, and this is just by construction, that the dominant dimensions in the activity don't have to be the same as the dimensions of activity that are relevant for predicting the activity downstream. And this is the sort of question we'll be getting into. What is the relationship between the predictive dimensions and the dominant dimensions of activity? All right. So that's the sort of the philosophy and an outline of the approach. And now I'll step into the actual detailed analysis that led us to find this communication subspace between V1 and V2. So what we're going to do is make use of the fact that in the cortex, how neurons respond to the same sensory stimulus varies from trial to trial. So if we show exactly the same visual stimulus over and over again, each time we're going to get a slightly different response. What I'm showing you here in the top is real data. So each square here is the response of 1 unit. The grayscale value corresponds to the response of that unit on a particular trial. And I'm showing you here just 4 trials of responses to the presentation of the same sinusoidal grading. So we just show the same grading over and over again. And each time we show it, we get a different pattern of activity in this V1 population. Of course, this is also true in V2. Each time we present the stimulus, we get a different pattern of activity in V2. And what we want to do then is make use of the fact that these activity patterns vary from trial to trial to understand which activity patterns in V1 are most relevant for driving activity in V2. So we're going to do that initially with simple linear regression. We're going to count the number of spikes in each unit in V1. And we're going to fit a regression model that allows us to predict this population of V2 units as a function of the V1 units. Now we realized very early on that to do this type of analysis, or to be able to interpret it, we needed a comparison case. And so in fact what we do is we take the full V1 population and we hold out a subset of the V1 neurons outlined here in blue that we're also going to predict using the remaining V1 neurons. So in the terminology of the talk, what we have is a source population. We refer to this as source V1. This is roughly 70 V1 neurons. We're going to use them to predict 30, on the order of 30 target neurons, also within V1. And then we're going to use exactly the same activity to predict responses also in V2. So we have a within area model and we have a between area model. The target V1 population is chosen so that it has the same number of units in a particular recording session as the number of units we have in V2. And the firing rate of each one of these units in V1 is also chosen to match as closely as possible the firing rate in V2. So if I were just to give you the data from V1 and V2 for the target population and for V2, it would be very hard for you to tell which data set is which. Okay. So how well can we do in predicting activity in these two populations? That's shown here. So this is the performance of our regression model. On the x-axis is our ability to predict activity in the target V1 population. And in the y-axis is our ability to predict activity in V2. So you'll notice 2 things here. One is that the numbers here are modest, so we can predict about 20% of the variance in the target population using the source population. And we think from simulations that that's because we're subsampling the V1 neurons that are projecting to V2. So in reality, there are millions of cells in V1 projecting to V2. We're recording from a small fraction of them. And if you do some very simple calculations, what you find is that even in the case where the mapping between areas is strictly linear, the best you can hope to achieve are performance values that are similar to the ones I'm showing you here. So we don't think that this is because a linear regression model is inappropriate. It just reflects the fact that we're only able to record a small number or a small fraction of the input neurons. The second thing you'll notice is that our ability to predict responses in V2 is roughly the same as the responses in V1, so the points lie roughly on the diagonal. If anything, we're slightly better able to predict responses in V2 than we are in V1. And this is important because as we compare the models in the coming slides, we're going to be comparing models that work equally well. So the differences I'm going to show you can't arise because one model is simply performing better than the other one. All right. So what we've done so far is basic multivariate linear regression, and I want to give you a little bit of geometric intuition into what we've done. So here is a source population of 3 neurons again. And the colored lines here would indicate the regression vectors or the predictive dimensions for each one of the V2 neurons. And in the way we've done this so far, we allow each V2 neuron basically to choose, what we choose for it, the best set of weights that generate the best predictions. So the regression vectors could lie in any direction in the source population, and in principle could fully span the activity space of the V1 source population. We were interested in the possibility that this was not the case, and that in fact the regression vectors lie in a subspace of the V1 activity, in this case shown as a plane. So imagine rather than the regression vectors filling the source activity, they lie in a plane. And the reason that that would be interesting is it would mean that there would be fluctuations in the source population, those are the fluctuations along this dotted line, that would not generate any activity in the downstream network because they lie orthogonal to the plane defining the regression vectors. So we want to understand, do the regression vectors between V1 and V2 span the V1 source activity space, or do they lie in a subspace? And to do that, we're going to use a technique called reduced rank regression. It's a form of regression. It's very similar to linear regression. So this is the reduced rank regression model. We're going to predict responses Y, which are the V2 responses, using weights applied to an input population, which is V1. Exactly the same as the linear regression model. The only difference is the weights in this case are subject to a rank constraint. So we can define these weights to lie in a one-dimensional space, two-dimensional space, and so on by using different numbers of principal components of the predicted responses. So, and this is in fact a knob that we can vary. We can decide and assess how well do our regression models work as a function of the dimensionality of the mapping between the two areas. Okay? And so that's what I'm going to show you next. So let's start with the V1 models where we're using the source population to predict the target population. The little triangle here in the corner is the full linear regression model I showed you before. And the circles and line here indicate the performance of the reduced rank regression model as a function of the dimensionality of that model. So if we allow the regression model only to have one dimension, we take a big hit in performance. And to reach the performance of the full regression model, we need to allow something like 5 or 6 dimensional mapping between these two populations of neurons. Okay. So that's the situation for V1 models. What about the between area models? And that's shown here at the bottom. When we did this same exercise using the same population now to predict V2, what we found very consistently is that those models between V1 and V2 saturated performance at a very low dimensionality. In this case, a dimensionality of 2 is sufficient to reach the same performance value as the full regression model. I want to emphasize here that these numbers are not particularly meaningful. They reflect the amount of data we have and the size of the populations we're recording from. But remember again that the comparison between the top and the bottom is fair because it involves the same source population and the same size target population. So the difference between the top and the bottom is the meaningful part rather than the absolute value of the quantities on the x-axis. So that's the first conclusion. When we apply models between areas, we see a low-dimensional mapping compared to models applied to activity within a single area. So this is one data set. This is just to show you the effects in the entire data set in the paper. On the x-axis is the number of predictive dimensions we need for V1 models to reach the performance of the full model. And on the y-axis, the number of dimensions we need for V2 models. And you can see that in almost every single case, the V1 models are higher dimensional than the models between different cortical areas. Okay. So what we have here then is a dimensionality bottleneck for between area models that's not evident in the within area models. All right, so that's conclusion number one. So one reason that one might see this low dimensional mapping between V1 and V2 is that V2 activity perhaps is low dimensional itself. So imagine that all of the V2 activity lay in a small subspace, for example, on a plane, then it wouldn't be surprising that we could predict that activity with a low dimensional V2 model. If the target V1 population was relatively high dimensional, so here shown as this sphere, this stretched sphere, then it would of course be expected that the models explaining this activity would need to be higher dimensional than the models explaining the activity on the left. So what we're going to do next is assess what exactly is the dimensionality of the populations that we're trying to predict. And we're going to use that, we're going to assess that using factor analysis and ask how many dimensions do we need to capture activity in each of these populations. So factor analysis is very similar to principal component analysis, and it gives us these dominant dimensions that are going to summarize these two populations. So in this exercise, of course, we're going to ignore for the time being the source population and just focus on these two target populations and ask what is their relative dimensionality. So that's shown for you here. The x-axis is the dimensionality of the target population, which is the number of factors or dimensions we need to describe them. Target V1 in blue, V2 in red. And you can see that the distributions are highly overlapped. If anything, V2 is slightly higher dimensional than V1. And so the fact that the models from the source V1 to V2 are lower dimensional can't be explained by a dimensionality constraint within V2 itself. What I'm going to do next is bring in for you the y-axis on this plot, which is the mapping dimensionality that we discussed, that is the number of predictive dimensions that we need from the reduced rank regression model. So what you see for the target V1 population is the points lie very close to the diagonal. When we record from a population of V1 neurons that has whose activity is 5-dimensional, then we need a 5-dimensional model to explain their activity. If we record from a group of neurons whose activity is 2 to 3-dimensional, then we need a 2 to 3-dimensional model to explain their activity. But the situation for the V1, V2 models looks very, very different. Even when we have relatively high dimensional V2 activity, the number of dimensions that we need to predict that activity from V1 is relatively low dimensional. So this is what we refer to as a communication subspace, which is there's a subspace of activity within V1, which is relevant for predicting activity in this downstream area, V2. Okay. So let me walk you through one more sort of technical analysis, and then I'll begin sort of zooming out and explaining to you some of the implications of these findings. So the last analysis that I'd like to walk you through is trying to understand how the dimensions of activity that are relevant for predicting V2 relate to the dominant dimensions within the source V1 population. So is it that V2 is only interested in a few dimensions of V1 activity, perhaps because it's less sensitive and it's only driven by the dominant modes of fluctuation in V1, or is it instead that V2 is interested in something other than the dominant dimensions of activity within the source V1 population? So to assess that, we're going to compare two models. One is the reduced rank regression models that I discussed already. And we're going to compare their performance to something that we call factor regression. So these two are very similar in spirit, but they differ in one important detail. So in factor regression, on the right-hand side, what we're going to do is break up this regression problem into two stages. In the first stage, we completely forget about the target population. We only look at the source population, and in the source population, we're going to define the dominant modes of activity within the source V1 activity. Once we've defined those dimensions, we project all of the V1 responses into that subspace, and then we perform regression to predict V2. So both reduced rank regression and factor regression involve dimensionality reduction and regression. In reduced rank regression, those two things are all done together. We're going to choose our subspace to maximize the performance of the models. In factor regression, we choose our subspace to summarize the source population, or the dominant modes of activity, and we do the regression as an afterthought, as a second stage. Okay. And so we want to know, do these two methods, if these two methods perform similarly, that indicates that the dominant dimensions are those which are important for predicting V2, or target V1. All right, so this is what it looks like in data. These, starting again with the V1 models, the filled symbols show you the performance of the reduced rank regression model, as before, and the open symbols show you the performance of the factor regression model. What you can see is that the points look really, really similar. So if we have a one-dimensional model, it doesn't matter whether we choose that to maximize performance or capture the dominant modes of activity in the source population. Those models perform roughly equally well. Similarly for higher dimensional models. And that suggests that these two, the target V1 population is being driven by are the dominant modes of fluctuation within the source V1 population. For V1, V2, the situation, again, is really, really different. So here's the reduced rank regression model performance. And here's the performance as a function of the number of dominant dimensions. What you can see is we need to consider many dimensions of activity within the source V1 population to achieve the same performance we get from a small number of predictive dimensions. And this suggests that these two dimensions are actually quite different from each other. So again, this is one recording session, and shown here on the right is the summary for the full population. Plotted on the x-axis is the number of predictive dimensions, and plotted on the y-axis is the number of the dominant dimensions that we need to keep to match the performance for each one of these numbers of predictive dimensions. So for V1, V1, the points are close to the diagonal. For the V1, V2 model, you can see in every case, the red dots are above the blue dots. We need a large number of dominant dimensions to achieve the same performance as a small number of predictive dimensions. So again, V2 does not seem to be driven particularly by the dominant dimensions of activity within V1. Okay. So let me summarize sort of in a cartoon way these findings, and then I'll talk a little bit about their implications. So what I've shown you is the following. On the right-hand side are the sort of the V1 results. And what we find is that when we try and understand the mapping between source V1 activity and the target V1 population, that mapping is relatively high dimensional, shown here as sort of a big open pipe connecting these two populations. And that the fluctuations that the target V1 population is interested in are the dominant modes of activity, which are the darker blues in the source V1 activity. Between V1 and V2, we see something distinct. So the mapping between those populations is lower dimensional, so it's like a small pipe relaying activity between these two areas. And the dominant modes of activity, what V2 is interested in are not the dominant modes of activity, but something that's rotated or somewhat distinct from those modes within the source V1 activity. Okay. All right, so we have very sort of different views of the way communication occurs within an area and between areas. So why is this interesting? Why would we care that the communication between areas relies on a relatively low dimensional mapping compared to within area? So let me walk you through sort of the implications of this and how this might be a general mechanism and an important mechanism for achieving selective communication between areas, and also for dynamically regulating the communication between different areas on a moment-to-moment basis. So I'm going to show you it's an actual simulation, but it's really, really simple. So we're going to create a population of neurons, 3 neurons. The whole population has 3 neurons. And in this model, each one of these 3 neurons in the source area in black is going to project to two different downstream areas, a blue area, area A, and then red area, area B. So this is the mapping between the two areas. Every neuron projects to every neuron downstream with a different synaptic weight. We refer to this as a full interaction where there's no dimensionality constraint. So I think this is the way most of us, certainly the way I thought about this problem years ago. If you record activity in the source area, as that activity fluctuates in time, if these neurons have connections with different downstream targets, those fluctuating activity patterns are going to be relayed to both of these downstream areas. So every little change of activity in the source area causes some change in activity in both of the targets. And this is true for a full interaction or a high-dimensional interaction between areas. Let's look at the same scenario now when we have a communication subspace. So again, all neurons in the source area project to both of the target areas. So the connectivity looks the same, but we've just imposed a dimensionality. Hello?
Hi. So we got disconnected. You can share your screen again. Yes, that's good.
It's back?
Yes, it's back. We can see it.
Okay. All right. Let me hide this. All right. So communication subspace. So all-to-all connectivity, but through a low-dimensional mapping. When this is true, there are fluctuations in this activity, again shown by the squiggly black lines, that generate activity in one area, but these fluctuations will generate no activity at all in the area B, even though there are synaptic connections between these areas. And the reason for that is, in this case, we've constructed these fluctuations in the source activity so that they fall in the communication subspace relaying activity to area A, but not relaying activity to area B. Now, using exactly the same pattern of synaptic weights, we can change the activity in the source population a moment later, and now we can achieve selective communication with downstream area B and not affect activity in area A at all. So the idea here is by generating different patterns of activity in the source population, we can dynamically regulate signaling between the source population and different downstream networks. So this is a way to achieve selective communication and also a way to dynamically change which areas are communicating with which. So this is our proposal that the communication subspace idea may be a general mechanism for instantiating selective and dynamic communication between brain areas. And I'd like to take a moment to compare it to sort of the competing idea in the field, which is that selective and dynamic regulation of communication is achieved by brain oscillations. So this is a very prominent hypothesis in the field. In the visual system, it usually focuses on gamma oscillations, and Pascal Fries, in particular, is someone who's done a lot of work in this area. I'm seeing the question, are there echoes? Is there a problem with the audio?
No, the audio is good.
Okay. All right, so Pascal proposes that to achieve selective communication, what you want to do is establish oscillations in both the source and target area. So consider a group of neurons C in a higher visual area, and those neurons are receiving input from neurons in a lower area, neurons, a group A and a group B. The way to achieve communication between A and C is to establish a gamma oscillation in A, a gamma oscillation in C, and to make those oscillations coherent so that the spikes occurring in area A are traveling to area C when area C is actually active. So in both A and C, activity is fluctuating with these oscillations, and if the oscillations are aligned, then communication can occur. In contrast, the neurons in group B, if they have an oscillation of a different phase, those spikes will arrive in C at a time in which neurons in group C are not able to respond or to receive those inputs because they're inactive. Okay. And then by changing the coherence between different areas and different times, you could dynamically change which groups of neurons are communicating with which. So there is very good evidence for gamma oscillations in the cortex. We've worked a little bit on this as well. And there's also evidence that the gamma oscillations change depending on the animal state. But we would argue that there are really some basic sort of design principles that make this perhaps more likely to be an epiphenomenon rather than the way that you would want to design communication between brain areas. So one is that this type of mechanism has quite low temporal bandwidth. As you'll notice, neurons in area A can communicate with C at discrete times, separated by periods of time in which the brain areas can't communicate because both of them are sort of in this refractory state. And of course, if you regulate communication with low frequency oscillations, you can have large chunks of time when brain areas can't communicate effectively because of this refractory period. In contrast, in the communication subspace, there's no temporal constraints. As quickly as we can change the activity in the source population, we can map activity onto one communication subspace or the other. And the reason for that is because here communication specificity is achieved by the pattern of activity across neurons as opposed to the pattern of activity across time, as in the oscillation case. So that's one sort of design advantage of the communication subspace. Another one is that this type of mechanism is prone to high interference between channels. So in this case, this might work very nicely because you have two inputs and you're trying to select one and not the other. Instead, you have 100 inputs and you wanted to select a subset. You wanted to listen to 20 of those inputs and not to the other 80. In that case, it can become quite difficult to establish the right oscillations among the different groups of neurons and prevent slop in the timing of the oscillations from allowing signals from one group of neurons to bleed into the communication in the other group of neurons. So there's sort of a finite number of different distinct phases, meaningful phases that you can offset that would allow for selective communication. In contrast, in the communication subspace idea, there are many different subspaces that we can fit in the source population that would allow us to communicate with different downstream targets with no interference, like in the simulation that I showed you in the previous slide. So I think certainly in my view, this is not a settled question. I'm proposing here the advantages of thinking about this through the communication subspace framework. But there are robust data supporting both of these schools of thought. And I think we're sort of in the early days of trying to disambiguate which are really the dominant sort of principles for determining how areas communicate. In an opinion piece that we published last year, we laid out some of these issues, and we argued that to understand between area communication, we need to start thinking not at sort of just documenting phenomenology in the brain, but thinking from a computational perspective of the design considerations that might be advantageous of particular schemes that people are observing in different parts of the brain. So here are a bunch of different considerations one might have. I mentioned the interference consideration and the bandwidth consideration. And there are others, how this different mechanisms might scale as a function of the number of different areas that are interacting with each other, how quickly we can reconfigure the network, how these different schemes for regulating communication might interact with computation. And so we're arguing in this piece for those interested that these are the sorts of the design considerations that we need to take into account when we evaluate the different sorts of communication schemes that people are proposing based on data. Okay, so in the last 10 minutes or so, I want to just show you sort of where we're going with this way of thinking and present to you some just published work on feed-forward and feedback signaling, and also show you some ongoing work in my lab that looks at the relationship between the communication subspace framework and behavioral variables. So for this last 3 or 4 slides, we're going to switch the pairs of areas we're recording from, still in the visual system, but rather than V1 and V2, we're going to be looking at V1 and V4. These are recordings performed by Anna Jasper, a postdoc in my lab. Anna trained animals to fixate and also to do perceptual tasks. And in trained animals, we then implant a bunch of Utah arrays, 2 96-channel arrays in V1 and 2 48-channel arrays in V4. So we're recording from close to 300 channels across these 2 stages of the visual system. through a little bit of education and a lot of luck, we are able to place these Utah arrays in a way that they, again, sample from groups of neurons that represent similar portions of the visual field. That's shown for you here. So the red circles are the receptive field outlines for neurons in V4, the blue for V1. You can see the two different V1 arrays and the two V4 arrays, but some of our arrays have, again, very nicely aligned spatial receptive fields. So the first thing that Anna was see whether this communication subspace that we saw between V1 and V2 in an anesthetized animal also occurs between V1 and V4 in awake animals, and the answer is yes. So this is the plot that I showed you before. On the x-axis is the dimensionality of the population we're trying to predict. On the y-axis is the mapping dimensionality. You can see that for each target dimensionality, the red points are consistently below the blue points. That is, the V1 models are largely constrained by the dimensionality of the target population, whereas for V1, V4 models, we have a low dimensional mapping from V1 to V4. And I'll just point out that several other groups have now seen communication subspaces between other pairings of areas. Marlene Cohen's group between area MT and the superior colliculus, and two groups that we're aware of, one from the Allen Institute and another through personal communication, have seen communication subspaces between different visual areas of the mouse cortex. So it seems it's early to say that this is a general principle, but at least it's not specific just to V1, V2, or V1, V4 in the monkey. Okay, so we have a communication subspace between V1 and V4. And now we want to make use of the fact that we have this subspace to explore feed-forward and feedback signaling between these areas. So I've emphasized so far models from V1 to V2 or to V4, but information doesn't just flow from early visual cortex to higher visual cortex. It also flows in the opposite direction from V4 or V2 back to V1. And we want to understand how are those signals being relayed in the feedback direction. So to assess that, we're going to do an analysis very, very similar to the type of analysis that I've been telling you about so far. It's called, in this case, canonical correlation analysis. Again, it's a dimensionality reduction approach, but rather than using one population to predict the other, it treats the two areas symmetrically. So specifically, what canonical correlation analysis does is it picks a dimension in V1 and a dimension in V4, such that when we project activity onto that dimension, the activity in the two areas is maximally correlated. Okay? So again, a dimensionality reduction approach, but we're looking for dimensions of activity simultaneously in the two areas that provide us the strongest correlation of activity between the areas. And we're going to apply this analysis between V1 and V4 as a function of time after we turn on a visual stimulus, and also as a function of time lag by varying the time window that we're counting spikes in, both in V1 and V4. All right, and so this is what that looks like. What I'm showing you here is the correlation between V1 and V4 along the first dimension identified by canonical correlation analysis. And I'm showing you that as a function of the delay of activity between the two areas. Sorry, so in the red trace, you see activity early in the trial, about 75 milliseconds after we turn on the stimulus. And you can see that the correlations peak for positive delays, which is V4 following V1. This is very consistent then with a feed-forward interaction between these areas, where activity in V4 is correlated with activity in V1 that occurred earlier in time. Later in the trial, about 125 milliseconds and later, the correlation pattern shifts so that V4 actually leads V1. V4 activity is going to be correlated with V1 activity that occurs in the future, consistent with the flow of information from V4 back to V1. So applying canonical correlation, what we see is a shift from feed forward to feedback as a stimulus persists. This is one example session, and this shows you the consistency across sessions. And this paper appeared, I think now, 2 days ago in Nature Communication. Early on in the trial, feed-forward communication, followed by a feedback-dominated communication late in the trial. So this by itself is maybe not too surprising. Lots of pieces of evidence suggest that the visual system works in this way. Of course, the first patterns of activity have to be feed-forward. V4 needs input from V1 to function. And there's evidence already in the literature that later on in the stimulus, feedback connections become more important. What was more interesting, I think, in this work than the shift from feed-forward signaling to feedback is that these two forms of signaling relied on different patterns of activity in V1 and V4. And that's shown for you here in this figure on the right. So this is actually data. What I'm showing you here is the ability of dimensions identified in either the early or the late epoch to capture correlations on either the early or the late epoch. So if we apply canonical correlation early in the trial and we ask how well in held out data can we capture correlations in the early epoch, we get a normalized performance of 1. The raw performance value is the one that you see here. When we use these same dimensions, these same vectors in V1 and V4 and ask how much correlation do we capture late in the epoch, we find that the correlations are much, much weaker. Similarly, if we use activity late in the epoch to identify dimensions, that works well late in the trial. But those dimensions of activity are very close to relay early attribution here. So what this means is that there are different patterns of activity that are involved in feed-forward and feedback signaling between these pairs of areas. And this also means, we hope, and this is the direction we're going, that we can use isolation recordings and this type of analysis to disambiguate the feed-forward and feedback signaling between these areas as a function, for example, of stimulus drive or what the animal is doing behaviorally. So for those of you interested, again, the paper is now online at Nature Communication. So the final thing, the final data slide is just to relate some of these communication subspace ideas to behavior. Everything that I've focused on so far is sort of basic visual processing where the animal is either anesthetized or staring at a screen and we're presenting visual stimuli. We're also very interested in understanding how communication subspace relates to behavioral goals and decision signals in the visual system. So again, Anna Jasper has trained an animal to perform a perceptual decision-making task. It's a match-to-sample task, so the animal fixates. 3 stimuli up here. One is a reference stimulus, and then there are 2 choices. And the animal has to decide which of the 2 stimuli is more similar to the reference stimulus, and then make a saccade to indicate its decision. We chose this task because the monkeys actually can perform this task for arbitrary stimuli. So in this case, we're using gratings. But in the actual recordings, we use not only gratings, but textures with different types of stimulus statistics and so on. So we can have the animals sort of doing different variants of a perceptual decision-making task in the context of the same sort of overall task structure. In these recordings, we wanted to know, the only piece of data I'll show you from this so far, is how the animal's decisions relate to these communication subspaces relating activity in V1 and V4. So the top plots here show you how well we can predict the animal's decision on each trial. So we have these population recordings in V1 and V4. We perform basic linear decoding. The performance of the linear decoder as a function of time is shown at the top. The gray is when the stimulus is on the screen. It's this epoch. And what we see in both V1 and V4 is the appearance of choice signals. That is, the responses in these 2 areas allow us to predict on held out data what the animal's choice was on a particular trial. The performance is modest in V1, about 55%. In V2, it's better, about 65%, and in some sessions, it's over 70%. So this is not too surprising based on our own work and other work. Of course, there's choice signals in both V1 and V4. Now the question is, how do these choice signals relate to the communication subspace that relates activity between V1 and V4? So Anna defined the communication subspace using the type of analysis I showed you before between V1 and V4. She then projected the activity in V1 and V4 into that communication subspace and asked again, how well can I predict the animal's choice within the communication subspace? And what's shown here in these different colored lines is how well you can do as a function of the dimensionality of the communication subspace. So you'll notice there's some stark difference between V1 and V4. In V1, you can achieve all of the achievable performance within V1 using a very low, really the first dimension of the communication subspace, whereas in V4, only a portion of the choice information falls in that low dimensional communication subspace. And what this suggests to us is that there's robust choice information in V4, which is not related to choice activity in V1, but all of the choice activity in V1 can be traced to the communication subspace relaying activity between V1 and V4. So this suggests that the choice signals in V1 may come from V4, whereas V4 has choice signals within that area which it is not sharing with V1 through the communication subspace. Okay. So to conclude, what I've tried to argue for today is the following. First, that studying inter-aerial communication should involve recording population responses and analyzing the responses, information coded by large groups of neuronal populations in different brain areas, and to understand inter-aerial communication, we really need to look at this through the lens of population-population interactions. I've shown you that inter-aerial interactions, both between V1 and V2 and V1 and V4, occur through a communication subspace. The concept of a communication subspace would allow for flexible and selective inter-aerial signaling. And I've showed you through this recent work that there are distinct subspaces for feed-forward and feedback signaling. So going forward, we're going to be using this sort of framework to study inter-aerial interactions, using neuronal population responses to understand how feed-forward and feedback signaling contribute to cortical function. And finally, I would just like to acknowledge the people who've actually done this work. So Anna Jasper is the postdoc who did the awake recordings, Amin Zanvakli, the anesthetized recordings. Elliot, Aravind, and Allison are doing communication projects currently in the lab that I didn't have time to talk about. And all of the work that I presented today involved this joint collaboration with Christian and Byron and talented students, Joao and a current student working on this, Everin Gokchan. And with that, I'd be very happy to take any questions.
Okay, thank you so much, Adam, for the great and informative talk. So we are almost out of time for the main hall, but we have a panel that we ended up closer interaction with you for the next 30 minutes, if you agree.
Absolutely.
So yeah, I'll just post the link here. So if you click on it, and you can just use the same username password to come to this room, and I'll see you in the same room.
Okay.
So people who want to have interactions with Adam, please log in to the panel.